Hi there, and welcome to this video on GCSE Biology for the AQA Specification, focusing on transport in cells and in particular osmosis. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started.
Hello and welcome to this tutorial on osmosis. So in our previous tutorial we had a look at diffusion, and in the next tutorial we're going to be having a look at active transport. So this is all about transport in cells, and osmosis is just another version of transport in a cell. So we'll be having a look at the basics of osmosis, various calculations, and if you just read through the specification points here, that will make it really clear to you exactly what you need to know. So perhaps you could pause it at this point and have a read through. So first of all, let's take a look at the definition of osmosis. So osmosis is the net movement of water from an area of high concentration to low concentration. So it's really important that you realise it only occurs in the movement of water, and it occurs only across a partially permeable membrane. So there's some really important points you've got to remember here. So looking at the definition, osmosis is the net movement of water, that's got to be in your definition, from an area of high to low concentration. So that's very similar to diffusion. It's moving down its concentration gradient, remember? And it's occurring across a partially permeable membrane. So if you're asked to define this in the exam, you would say, osmosis is the net movement of water from an area of high concentration to low concentration across a partially permeable membrane. Okay, that's really, really important. Your examiners are really likely to give you marks for water, high to low concentration, and partially permeable membrane. Those would be your 3 easy marks right there. And again, just in diagrammatic form, you can see there's more water molecules on this side of the partially permeable membrane. This would be the partially permeable membrane here. And fewer more water molecules on this side, so they're moving down their concentration gradient. Okay? Now, as I said, it's really important that you remember that the membrane is partially permeable in osmosis. That's really important. You're going to get a mark for that if you write that in your definition. Now, on the last slide, we defined osmosis as moving from water molecules moving from a high to a low water concentration. But another way of referring to the idea of concentration of water is referring to it as water potential. So a high concentration of water is a high water potential, and a low concentration of water is a low water potential. So how I think of it in my head is you can just use those words interchangeably, but for the purpose of the exam, instead of saying high water concentration, you would say high water potential, okay? And same with low water potential. And it's moving down a potential gradient, a water potential gradient. Now, it's really important that you know the distinction between osmosis and diffusion. So they are both, there are many similarities between both. So both involve a movement down a concentration gradient. Both are passive movements because they do not require energy. And, oh, that's it. I think those are the 2 big similarities, I would say. But osmosis only involves water, whereas diffusion can involve any sort of substance. Okay, so diffusion, when you spray some perfume in your room, your mum may smell it on the other side of your room. So that's because this substance has diffused across the room and she can now pick up the smell even though she's standing 10 metres away from you. And osmosis, on the other hand, is only occurring in water. Okay, so you see the difference there. Diffusion can occur in gas or liquid. Now, moving on to our second specification point, we're going to have a look at some mathsy things. Now, if we want to measure the rate of water uptake, now let's have a look at this scenario. So we have an apple that weighs 50 grams. We are leaving it perhaps in water for 30 minutes, and we measure it after 30. We measure its mass after 30 minutes, and we see it's 62 grams. Okay. So we want to measure the rate of water uptake. So this is a really simple calculation. You've just got to think it through logically. So first of all, let's look at the change in mass over time. Okay. So that would be 62 grams minus 50 grams. So you would, oopsie, changed colour. 62 minus 50 grams. Now, if I were you, I'd just write everything out, all you're working out, so that the examiner can see every little stage, so that even if you go wrong and you write 10 grams here, they will know that actually you've used the right logic and they can still award you the mark. So, oh dear, I keep changing colour. So 62 grams minus 50 grams is 12 grams. Yeah? So this is the change in mass over time. We're saying that is increasing by 12 grams in mass. Now, we want to find out the rate of water uptake. So that's always the change in whatever you're measuring over time. Now we know it's 30 minutes, right? We've been told in the question we've left the apple in water for 30 minutes. So you're saying that there's in 30 minutes there is a change in 12 grams. So in 12 grams, 12 grams. And then I kind of like to write equals 30 minutes. And what I mean by that is that in the space of 30 minutes we have a change of 12 grams, okay? That's all I mean. And if we want to measure a rate, we've got to get the time down to a basic unit. So either down to 1 minute or to 1 hour. So we can measure the rate of water uptake per minute or per hour. So if we want to measure it per minute, so per minute, okay, what we would do is we would write equals 1 minute, and we've then got to work out how do we get from 30 minutes to 1 minute. We divide by 30, right? So we've got to do the same to the other side of the equation. So 12 divided by 30 would give our rate of water uptake per minute. Okay, so what I would then do is do 12 grams over 30. Okay, so do you see my logic there? To go from 30 minutes to 1 minute, I'm dividing by 30. So therefore, to go from 12 grams to how much is uptaken per minute, I would also have to divide by 30. That's my logic. That's it. Really, really simple. And 12 grams divided by 30 is equal to 0.4 grams per minute. Okay, so that would be your rate, and that's how you write it. It would be grams per minute. Okay? Now I said earlier when we're calculating a rate, we can either express it in terms of per minute or per hour. Now it's really, really simple to go from per minute to per hour. So if I were to just put a little star here and go up here now. So if we want to write it in per hour terms, so we've already said 0.4 grams. A 0.4 gram increase occurs in 1 minute. So in 1 hour, which is 60 minutes, what's going to be our increase? So why don't you pause the video and try and work it out. Use the same logic that I used here with these yellow arrows. Have a look at how we go from 1 to 60 minutes, and we've then got to do the same to the other side. So just pause the video and try that. Okie dokie, so if you've had a try at that, then you can see that if we go from 1 to 60 minutes, we're multiplying by 60. Okay? So we're going to have to do the exact same to the other side, because we want to convert our per minute to per hour. So 0.4 grams times 60 would be 24 grams. So therefore we're saying that the rate per hour would be 24 grams water uptake per hour. Okay? So that's really, really simple. All I've done to go from here to here is multiply by 60. And there's so many different ways you can do this. It really depends on how your brain works. So in my head, it's more logical to do it this way, but maybe in your head, you might have seen a little catch here, and you can see that we're getting an increase of 12 grams in 30 minutes. So if we want to measure the rate of water uptake per hour, 30 times 2 is 60 minutes, so that gives you your hour. So therefore 12 grams times 2 is 24 grams. So that's another way to get to this answer. So you can see how you can work it out in multiple different ways. It just depends on your logic as you do it. Okay, so we'll now look at percentages. So percentages can be used to easily compare values from the start and finish of osmosis. And this will therefore allow you to compare values between different specimens, for example. So let's have a look at an example here. So we're going to have a look at the same data from before. So we have our same apple going from 50 grams to 62 grams. We want to calculate the percentage change. So to calculate the percentage change, you always have to do how I remember it. This is my little equation for remembering it. Is the new kind of value minus the old value over the old value multiplied by 100, because this part here is your change, so this would be your change in value, and your old mass is the comparison of that change in value, okay, and then you multiply it by 100 to get your percentage. So in this case it would be 62 minus 50 over 50, because that's your new value, that's your old value, that's your old value, multiplied by 100, which is 12 over 50 multiplied by 100, which is 0.24 times 100, which is 24%. Okay, so that's your percentage change. And if that came out as a negative number, all that would mean is you're getting a percentage decrease. So let's say the apple started off at 62 grams and it lost water, came down to 50 grams after 30 minutes. These values would be the opposite way round. You'd therefore get negative 12, and therefore you would get a negative percentage change in mass, okay? So if you get a negative, you know why. It's just because there's been a loss. And let's say you were to get a negative here for this answer. A really good thing to do is to go back and think logically through it and think, okay, I've got a negative percentage change. Does that agree with the question? And if I were to have had a negative percentage change here, I would know that I've gone wrong, because the apple's increased in mass, not decreased. So we are expecting a positive number for the percentage. Okie dokie. So this is all the working out that I just did for you. So this graph also shows percentage change, but in a graphical format. So we're looking at the concentration of sucrose in a solution on the x-axis, and we're looking at the percentage change in mass of a cell on the y-axis, or not necessarily a cell, it can be anything, a slice of potato, let's say. And so, a few points to note here. So where the graph crosses the x-axis, x-axis, we're saying there's a 0% change in mass, which means that a sucrose concentration of 0.25, whatever these units are, is going to be equivalent to the sucrose concentration in the object whose mass is changing. Because remember, if your concentrations are equal, okay, then you're going to not have a concentration gradient, and therefore osmosis is going to be very minimal. So what I mean here is that, let's say you have your cell with its partially permeable membrane. Now you have more sucrose molecules. If you have more sucrose molecules on the, so if I call this the cell, and this part is out, this part here is outside. Now if we have more sucrose molecules, okay, on the outside, they're going to move into the cell by osmosis down the water potential gradient. Well, down the concentration gradient into the cell. Also, if you have more sucrose molecules on the outside of the cell, it means that you actually have fewer water molecules, right? So the water potential is going to be lower, and therefore water will move out of the cell, down its water potential gradient, okay? So here, you can see at this stage where the sucrose concentration is higher outside of the cell, we're going to have more of these sucrose molecules, therefore the water potential is going to be lower, and therefore we're going to get more water moving out of the cell, and therefore our percentage change in mass is going to be negative because it's losing mass because water is moving out of the cell. Whereas at this point here, the sucrose concentration is equivalent inside and outside of the cell, so there's going to be less water movement. So notice we're talking about water movement because here we're looking at the change in water moving across this partially permeable membrane. But here, where the sucrose concentration outside the cell is perhaps lower, so we can see that here it's lower on the graph. So there's going to be fewer sucrose molecules, and therefore comparatively there's going to be more water molecules. So we're not going to get movement in that direction, we're going to get movement in this direction, okay, because it's moving from a high to a low water potential, down its water potential gradient. Okay, does that make sense? So you can see how this graph might be confusing if you don't really annotate it, but as soon as I drew my little diagram here, it made more sense to me, it probably made more sense to you. So in the exam, don't be afraid to just do little scribbles on the side of your examination paper, because then you can work out exactly what's going on. Because sometimes it's quite confusing to say, oh, well, when the sucrose concentration is higher, we've got negative percentage change in mass. Well, what does that actually mean? Well, we drew it out and we realised that when the sucrose concentration is higher, the water potential is lower, therefore we get water movement by osmosis down its potential gradient from high water potential to low water potential across a partially permeable membrane. Therefore we get this loss of mass in the cell, and we therefore see a negative percentage change in mass, and vice versa for when we have a lower sucrose concentration outside the cell. Okay? So well done for today. Quite a nitty-gritty tutorial, lots of little points there, so do go back and watch it through again, especially the mathsy parts. And I will see you for our next tutorial on active transport.
Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.